So as I mentioned to Andrea, that I didn't plan it this way, so all the three speakers this morning are from Boston. And the third speaker is Selim Ünlü from Boston University, who's going to tell us about digital detection of molecules, a new frontier in biomarker analysis. Thank you.
Good morning. It's great to be here, and I'd like to thank the Standing Committee for letting me participate in this exciting event. It was fun, and I can see how much work it is. And I'm an engineer. My presentation will be more on the technologies for single molecule detection. And I'm coming from Boston University. There's an aerial view of Commonwealth Avenue. I'm just going to guess this is the button. Oh, the big green one. Yeah, well, it's the largest button on the thing that I missed. Anyway, I'm an electrical engineer, and my understanding of the biology world is from an engineering perspective. And the technologies we work on are related to using optical interferometry to enhance the visibility of nanoparticles. Those can be viruses, other biological nanoparticles like exosomes, and also labels that you can attach to individual molecules like nucleic acids or proteins. Then you can do single molecule detection. So I will give my philosophical approach to analog versus digital, and talk about the success of fluorescence microarrays and what the limitations are, and how the first attempts to do single molecule detection was based on fluorescence labeling. And then I will present a variety of different methods based on the approach, interrogating small volumes, reducing the area you're analyzing at a time, and how we can achieve single molecule sensitivity by compartmentalizing the solution into smaller volumes, or using high quality factor resonators, and finally what we've been working on, that is using non-fluorescent labels. So as part of the organizing committee, I really wanted David Waltz to be here giving this talk on the technologies for single molecule detection. And I just wanted to quote from his paper in analytical chemistry. He says, I removed the perhaps in this beginning of the sentence. Future of all analytical measurements will be molecule counting. And the bottom line is it cannot get any better. So when you look at this picture, the difference between the analog signal, where you're trying to measure how much signal you have, you can make a count how many molecules you have. The big difference between the two methodologies is it is much easier to make a decision if something exists or doesn't exist. Right? It's the presence is much easier to decide. It's a 1-0 binary decision rather than trying to do a precise measurement of the quantity. So the analog signal, as you see in this ELISA plate, you have the density of the substrate dye, and you try to make a measurement based on how opaque each microwell is. I liken that to an analog recording medium where the music is recorded as the height of the grooves on a vinyl disc. So if many of you would remember those days where we have, I mean, in a different audience, I said none of you will remember this because all students. Here I can say, you know, some majority of you will probably recognize these vinyl discs. Anyway, so if you remember, one of the most expensive components of your music system would be the player, because how you transduce the signal from this analog recording will be the limiting factor, and how you can reproduce the recording later on. So in 1983, Sony came up with the compact disc player, and it was $900. You know, from $83, it's probably several $1,000s today's money. And it was very expensive, but it had the path towards being very inexpensive later on. Only 3 years later, in my dorm room at Illinois, I was able to have a compact disc player to reproduce music as good as anybody else. Even if you're the richest person on earth, you could not transduce the recording any better than a compact disc player now you can buy for maybe, there are no compact disc players anymore. All the digital recording is on computers, on discs, well, on the cloud, right? Anyway, so the SIMOA, the single molecule analyzer from Quanterix is shown in this picture, and I liken that to the first generation of digital recording, the compact disc players. So if you go back in time again, and this is, you may recognize him, this is Muhammad Ali. You need to be rich and famous to be able to have vinyl recording portable, right? I have only seen one vinyl player in a car in my lifetime. And right now, we can carry the music around in a compact disc player, not even. And the transition in digital detection will enable that these technologies will be available at low cost, and it will be pervasively available. That's my prediction. So when we go back in terms of the motivation, the success of fluorescence DNA microarrays is inspiring, because they were able to put on many thousands of unique probes, and then you incubate with the either DNA or RNA targets, hybridize, stain with a fluorescent reporter, then you can measure the fluorescence of each spot. You do thousands and thousands of measurements simultaneously, and you learn something about what's in the solution. The problem, however, is the sensitivity of such a technique that is limited by how well you can read out the fluorescence signal. To be above the noise level for a typical fluorescence reader, you would need to be at several to 10 fluorophores per micron square. However, your spots are not 1 micron square, and there's reasons for them not to be 1 micron square. And when you look at a 10,000 micron square spot, we're talking about requiring as many as 100,000 probe molecules or captured fluorescent molecules per spot to be above the noise level. And that not only limits the sensitivity, but also reduces the dynamic range of where you can quantify the concentration of the target molecules. So typically, dynamic range is 2 or 3 orders of magnitude, and the sensitivity is in picomolar quantities. Well, there is plenty of room at the bottom. I wish I was the first one to say that, but that's a very nice word, a nice concept. As you see in this chart, you can actually get many more orders of magnitude in your dynamic range by going down to a single molecule per spot if you could detect individual molecules regardless of where they are located in the solution. And then you can improve the sensitivity and dynamic range by 4 orders of magnitude. So this hasn't been done until relatively recently. I'll give you some historical perspective to sort of set the stage on how we have the technologies today that can now really do this. So, to achieve the sensitivity limit, we can do different things. As I mentioned in the beginning, we can interrogate small volumes or small areas, and there are different ways of doing it. One is just increase the magnification of your optical readout, look at a much smaller area with an oil immersion objective with 1.3 Na, and then you can start doing single molecule counting. Or you can use total internal reflection to reduce the volume that you're interrogating. And one of the successful applications of this is what's called the zero-mode waveguides. I'll tell you a little bit about it. Or you can basically take your solution and break it into tiny little pieces, to droplets, or into microfluidic compartments, or in microwells, and then you can do 1-0 measurements. Again, you can now digitize your measurement. It's very much like a compact disc. You're trying to make a decision between, is there a fluorescent presence or no, as opposed to trying to measure how much there is. Since I'm an electrical engineer who worked on optics, I have to mention high-Q optical resonators as a potential way of doing it, and I'll tell you about their limitations. Finally, I'll talk about the area of research we work on. There are other groups as well, on labeling the single molecules with non-fluorescent labels. In our case, we're using an interferometric method to read out individual single gold nanorods. So now back to fluorescence microarrays. One can imagine that if you increase the magnification of your objective, then you can look at smaller and smaller areas with higher and higher collection efficiency and get single molecule readout. The first work was 1976. Hirschfeld was able to detect individual antibodies. He estimated that there are 80 to 100 fluorescent molecules tagging each antibody, and he saw very clear signals. Fast forward 30 years or so, the group in Salzburg was able to show single molecule sensitivity reading a fluorescent microarray, and they demonstrated that you can improve the concentration sensitivity from a picomolar, a couple of orders of magnitude, down to a 10 femtomolar range. So at the University of Illinois, they used the total internal reflection fluorescence techniques to interrogate a small volume by benefiting from the evanescent field, illuminating only the very close proximity of the surface, and therefore they could do single molecule counting. So all of these methods are actually quite difficult and require sophisticated instruments and their throughput is rather limited because you can interrogate a very small area at a time. One of the significant innovations was done by the Cornell group by using what they called zero-mode waveguides. Basically, you have light impinging on a surface, and you build a small well in which light will no longer be able to propagate. So it will be evanescent, it will be confined in all 3 dimensions, it will be basically stuck on the surface. And when you have molecules captured on many of these dots, you can see how big an array we're talking about. There are individual zero-mode waveguides in these arrays, and then you have arrays of arrays, and you can do very many measurements simultaneously without necessarily trying to focus your light, because the light is already, by the virtue of the chip, is focused in the individual parts, and then you can read out many, many events simultaneously. So Pacific Biosciences was a company that was formed out of this effort, and they've done real-time DNA sequencing, and a couple of their recent successes included basically determining the origin of the cholera outbreak in Haiti, and they also determined and demonstrated that the E. coli outbreak in Germany was the E. coli proteins had acquired the gene to produce the Shiga toxin by lateral gene transfer. And these papers were published in New England Journal of Medicine. And I found out that Illumina now is acquiring PacBio, Pacific Bio, for over $1 billion, and that's going to go through next year. So when you go back in time and try to determine when the first single molecule experiments in biology were done, that is going back to a Stanford professor, Rotman. And what he was able to do is basically the second method, reducing the volume by taking a very tiny sample. In his case, it wasn't that tiny. You can see that the scale bar is about 10 microns there. And a solution containing the beta galactosidase. See, I'm not a biologist. Anyway, so, and a fluorescent substrate was sprayed over a silicon oil, and after several hours, these emulsions formed and was able to determine individual events of having molecules by looking at which of the droplets are glowing. Now, again, fast forward, we now have methods. This work is from Harvard. We have now methods to generate millions of little tiny droplets in picoliter volumes. When you think about concentration for a microliter, you have 1 million times higher concentration if the same amount of molecules, same number of molecules exist in a picoliter volume. So the idea of reducing the volume to tiny pieces and then individually interrogating them is allowing you to do single molecule detection. Now, with the droplet formation for droplet microfluidics, one of the other advantages is that you can inject different solutions into the same droplet. As shown here, you can basically generate them, store them in large quantities, and then inject different types of dyes, different recognition molecules into the same droplet. So on a similar fashion, there is the concept of the slip chip. It's a nanoliter volume immunoassay, in which case you have basically two sliding plates, and in these sliding plates you can see you have a volume that you do your reactions, and you can slip the top substrate by, well, a top film in which you have the sample, different wash buffers, and the dye substrate. So you can do these assays one small volume at a time, and you can have many of them done at the same time by having a number of parallel channels. So you slide the top piece and you do many, many reactions simultaneously. Actually, I like this technique very much. It's been also commercialized, but it is simple enough. So if you just follow these YouTube videos, you can see that they can have little children, school children, actually run such experiments. So perhaps one of the most successful single protein measurements was done in microwell plates. Microwells. So in this case, you incubate your target solution with a bead, which has the capture antibodies, and then incubate with a biotinylated detection antibody. You capture that if the antigen is captured. And again, we get this beta galactosidase molecule, and that will change the color. But instead of trying to do this in the bulk solution, you take your sample and spread it into hundreds of thousands of microwells and read out from the bottom with a fiber bundle array, and you can do single molecule counting. I'm not sure if you're able to see this. A white light image shows where you have the beads, and the fluorescence image shows which beads actually have the captured molecule. So by controlling the density of the beads, you can reduce it to a single event per microwell, so you do truly digital detection. This is the technology commercialized by Quanterix, and it's called single molecule assay or single molecule analysis. I like their advertisement picture, which shows an iceberg. Because of the sensitivity of analog assays, you're limited to the tip of the iceberg, but now with the digital assays available, as you see here, as opposed to a plate reader being confined to maybe one or two logs of a signal, you have now 4 more logs of signal available to you in terms of the concentration of the molecules in the solution or the number of counts. And they have identified a variety of different areas for impact: oncology, neurology, cardiology, inflammatory diseases, infectious diseases. And the typical sensitivities they can achieve are in the order of attomolar concentration or femtograms per milliliter concentration. So, as I said, as an engineer, I need to tell you about the resonators, high-Q resonators. One of the approaches to see how we can increase the signal from a small event to have a large observable signal is, you focus the light, or you can resonate the light so it is confined in a small volume. And this has been done by a variety of different techniques: nanohole arrays, photonic crystals, rings, toroids, disks, what have you. As this cartoonish picture shows from Fulmer's work a while back, that in a whispering gallery mode device, a high-Q resonator, by interrogating how light is stored in this resonator, you could determine the presence of a bound single molecule to the surface. One of the problems is you need many of them. You need hundreds of thousands of them to actually be able to interrogate a bulk solution. And the second thing is the signal wasn't good enough for a single molecule detection. They had to add additional plasmonic nanoparticles to the side of these type of resonators. I would urge you to read the mass transport limitations. This is a good paper with Manal's group, and making a stick, and the difficulty of having highly resonant, small interaction volume, or nanosensors that will produce a signal, a discernible signal in the presence of a single molecule, is the mass transport, right? Imagine that this microphone stem is your nanosensor, and this room is your solution. How likely is it that single molecule is going to come and find this? And what I want to talk about is a part of our work where we try to make the carpet the single molecule detector in the same volume. And using micro nanoparticles in biosensing is nothing new. It's been used attaching additional particles, mass tags, or nanoparticle tags, even in ELISA type of measurements. It's been well known. You can improve the signal significantly for optical resonators. You can use silver staining with a catalyst gold nanoparticle, or you can improve your electrical detection techniques attaching nanoparticles to your molecules. Now, why do we want to use nanoparticles and non-fluorescent light scattering labels? Well, first of all, it's not going to saturate. The more light you put on a gold nanoparticle, it will give back more light to you. And you can reduce the complexity of the instrument when you try to do the single molecule detection. And you can also do dynamic measurements. You're not limited by bleaching of the fluorescent dye. Obviously, if I can attach a micron-size bead, I can do the detection in a very simple way. The difficulty is the ability to see small non-fluorescent labels. So that's what we've been working on to demonstrate that this was actually a simple method. It was done by 2 seniors undergrads. When I came up with these ideas, I asked them, because we didn't have specific funding, so I asked George and Rahul if they can work on this as their senior design project. And well, give or take 8-9 years later, and $10 million spent, we're in a much better position today. But they were able to demonstrate the basic concepts as 2 undergrads working maybe 5 hours a week or 10 hours a week. And this illustrates how it works. So a single nanoparticle does not produce sufficient signal that you can see if it is in solution. But if you capture on a surface, you can benefit from reflection from the surface to provide a reference field, and you're interferometrically enhancing the signal you will observe from a nanoparticle. And on your camera, it will appear as a bright or dark dot. And from that, you can determine the size of the particle if you're interested in analyzing the particle. And George, after finishing his undergrad, he did his PhD, and now he started a company with another my group members. I'm not involved with any of these companies that I'm talking about, so there's no conflict for me. So, and what they do is they count size and phenotype, what they call the invisible individual exosomes, down to, I believe, 50 nanometers in diameter. So the technique, the interferometric reflectance imaging with single particle counting modality, can detect viruses and exosomes without labels, but it comes down to individual proteins. Other people using similar techniques can actually see individual proteins directly, but we're much more interested in practical techniques. For that reason, we label them with individual gold nanoparticles, and then they become visible. So I wanted to plug for one of my former students, Darren, is now working at Harvard, and Fulya is about to finish her PhD by the end of this year. So the significant improvements we made in this system is using gold nanorod labels and polarization of light to enhance the visibility of such particles. And instead of using a 100x objective or even a 50x objective, we use a 10x objective and still can see individual nanoparticles. And these nanoparticles are only 20 by 60 nanometers in size, so they do not affect the affinity of the individual molecular binding. So you can see how this assay works. You have the capture antibodies, and you capture the antigen and decorate with a gold nanorod conjugated antibody, and then you count. Or you count in this field of view, and you can do a multiplex measurement and get down to femtogram per milliliter concentration. So we can also, this is our paper published a couple of weeks ago in PNAS, we can also do this dynamically. Since there's no bleaching and the signature from these metallic particles are not changing over time, you can do dynamic counting of single molecules. And therefore, you can beat the reaction limit. At a given time, you have less than one molecule on the entire surface of the sensor. But over time, you can determine what is bound, what is left the surface, how much each label or molecule stayed on the surface, and therefore you can learn more about their dynamics and differentiate between a high affinity, low concentration versus low affinity, high concentration analyte. So just wanted to give you one example of what we are using the single molecule assays in our lab. We are working with an Italian collaborator at CNR Milano, and they do prepare the samples with rare mutations in wild-type molecules through their patented PCR technology, and we do the readout. We do the readout by capturing them on our surface and reading the presence of these gold nanorods. Just results as of yesterday, we're able to show that we can detect 2 different mutations in the presence of 100 times more wild-type DNA available in the same solution with very, very good contrast. This is one mutation and the presence of the other mutation. You can compare the signal to the background by looking at the size of these bars. And these pictures, the videos are showing a tiny fraction of a single spot to make that visible. Otherwise, when we're looking at these images, at a given time, we're looking at as many as 10 spots in the field of view. So, in conclusion, single molecule detection, counting individual molecules is the most accurate method for measuring the concentration in a solution. And you can do it quantitatively and with extreme sensitivity. And optical methods have emerged as the powerful tools. Everything I mentioned are optical methods. And in my opinion, and also I think I read in some of the papers by David Walt, there will be a direct complementarity between single cell work and single molecule work. Because in a single cell, you have the absolute number of molecules in tens or hundreds. So you need to have a single molecule counting methodology to actually study the molecular count of individual cells. And more than 1,000-fold sensitivity improvement, it should allow to expand to a much larger variety of proteins and nucleic acid markers. It could allow you to use maybe 10 times as many proteins for diagnostics that's currently available. I believe it's in the hundreds of different molecular markers. Protein molecular markers are FDA approved for diagnosis purposes. You can increase that to many thousands, given that you can improve the sensitivity. We can do earlier cancer detection, earlier infectious disease detection, identify new biomarkers for diagnostic screening and environmental monitoring, and liquid biopsies, precision diagnostics. These are the recent buzzwords. I think these will be all enabled by digital detection of biomolecules. Just wanted to show a picture of my group when it was in its heyday. Now it's more like 10 people. We're still continuing to work on interferometric enhancement, and I'd like to thank you all. And if we have any time between now and the break, I'll take some questions.
Yeah, thank you, Selim. So I think we are a little bit over. Is there one burning question? Otherwise, we'll take the other questions during the break. Okay, good. Thanks again for all the speakers in the morning session, and 15 minutes.
5 minutes.
Okay, so we'll be back in 5 minutes, I'm told, to catch up on time.
It says break, but it doesn't say coffee break. Is that coffee?